Okay, so welcome everyone to the Northwest RISC's first event of the year, our long-awaited webinar on Northwest weed biocontrol in changing environments. This event is a production of the Northwest Regional Invasive Species and Climate Change Management Network, which is itself a program of the Northwest Climate Adaptation Science Center. We're a partnership of researchers and practitioners who are dedicated to helping address challenges at the nexus of climate change and invasive species. And I'm Eva Kohlberg, the coordinator for the Northwest RISC. I'm joined today by 5 speakers, starting with Dr. Jenn Andreas, who will provide a brief overview of the basics of weed biocontrol. Then Dr. Chandra Moffatt, Fritzi Grevstad, and Brittany Barker will provide some examples of how climate can interact with different aspects of biocontrol systems. And Dr. Annette Evans will round us out with some findings from a global review of climate change impacts on biocontrol. Just some housekeeping notes before we get started, though. We are recording this and we'll post that on our website and YouTube. And if you are joining us live, you can put in your questions in our Q&A box. If you have specific tech issues or things that aren't quite questions for the whole audience, you can also chat us via the chat box. You can also, I think you have reactions enabled. If you don't, whoops. But also do stay tuned for the end of this when we'll be giving you a little bit more information on how to get involved with this topic and RISCC, our new Northwest Weed Biocontrol Working Group. So stay tuned for that information. But with that, I'm going to hand things over to Dr. Andreas, who'll give us some quick background on weed biocontrol.
Thanks, Eva. I'll just share my screen. Okay. My name is Jenn Andreas. I'm with Washington State University, and I'll begin today's webinar with a very brief introduction of biological control. Biological control, or biocontrol, is the intentional use of one living organism to control or suppress another organism. Most of the presentations in today's webinar will focus on biological control of invasive plants, where we typically use insects, mites, and pathogens as biocontrol agents. There are several types of biocontrol that I won't have time to go into today. For this webinar, we will focus on classical biocontrol, or importation biocontrol as it's sometimes referred. The practice of classical biocontrol is based off the theory that when plants arrive in their new range, they are released from the natural enemies that keep their populations in check in their native range. This allows them to spread unchecked and gives them a competitive advantage over native species. So with classical biocontrol, we go back to the weed's native range and we look for specialist natural enemies that not only cause significant damage to the plant, but are also host-specific and won't attack non-target species once they're released. After extensive testing and a regulatory process that ensures host specificity, we then reunite the plant species with its natural enemies with the goal of achieving the balance we see in its native range. This type of biocontrol is typically used for invasive plants and insects, and it relies on the closely co-evolved relationship between the target weed and its natural enemies to find suitable host-specific biocontrol agents. As I emphasized in the previous slide, ensuring host specificity is the cornerstone of the practice, and this is done through host specificity testing. A wide variety of tests are conducted on a candidate biocontrol agent to establish its host range. And this testing can involve 50 to over 150 closely related plant species before the host range is established. Any candidate biocontrol agent that poses a risk to native or crop species is then eliminated. Biocontrol researchers and practitioners are often asked whether it is a safe practice and whether biocontrol agents will attack other species once they're introduced into a new range. In weed biocontrol, there have been extensive reviews that address these questions, and we've learned by looking at historical data that the types of tests and the testing sequence has a very strong track record in identifying safe agents, and that we also have a 99% success rate of predicting non-target use. In a global study of 457 intentionally released biocontrol agents, Less than 1% or 3 cases threaten populations of non-target species, and none of these would be approved for release today under modern regulatory systems. Classical weed biocontrol is an equally valuable component of a robust integrated pest management program, which also includes herbicides, mechanical, and cultural control methods. It is often used in rangelands, natural areas, and can be beneficial in reducing weed populations that cause human health issues. We also see long-term and self-sustaining weed reductions, and it's very cost-effective. Understanding how biocontrol agents and their associated weeds will respond to changing climates is a relatively new area of study. We expect to see both challenges and opportunities. We know that biocontrol agents are highly adaptable, which should be a benefit with shifting populations and climates. Matching the phenology between the target weed and its biocontrol agents is critical for success, and some of our presenters will touch on this topic today to help guide a path forward. We also expect to see an increase in extreme weather events and have already observed that biocontrol agents can quickly move in after something like a devastating wildfire and help restore habitats to pre-fire conditions. With this webinar, we hope to share some preliminary considerations around biocontrol and changing environments and start building relationships to grow this area of study. With that, I'd like to hand it off to Chandra Moffat, a biocontrol researcher with Agriculture and Agri-Food Canada.
Okay, awesome. So I'm here today. I'm going to talk about some data that's been collected in my lab with Agriculture and Agri-Food Canada in British Columbia by a master's student, Andreas Barretto, who's here in the audience, talking about biocontrol efficacy of St. John's wort and how phenology, abundance, and chemistry vary under geoclimatic conditions and biological control. So St. John's wort is an invasive, highly toxic plant that has been introduced from Europe and is now a global invader. It's invasive in more than 20 countries around the world, including Canada, the United States, Australia, and a number of countries in South America. The reports of wild St. John's wort in North America date back to the early 1700s, and it was recognized as a problem because of its highly toxic foliage that endangers both livestock that have been raised in North America and around the world, and as well as native ungulate populations of herbivores. It proliferates very extensively in open, disturbed dryland habitats, and it's highly difficult to control via cultural, mechanical, or chemical means, making it a great candidate for biocontrol. Slide change. So, St. John's wort is Canada's oldest weed biological control program. There have been 5 agents introduced into Western North America, including the 2 species of chrysolina, which are defoliators, Chrysolina hypericum and Quadrigemina, the moth Aplocera plagiata, the aphid Aphis chloris, and the buprested beetle Agrilis hypericum. So not only has biocontrol of St. John's wort been highly successful in Canada, it's one of the world's most successful weed programs. These are very famous photos from the U.S. Geological Survey and USDA ARS that show St. John's wort infestations in California, both sort of at different stages of the invasion. So it's often said that this is before and after, but actually in the left image, the person standing, the biocontrol agent, the 2 species of chrysolina, have already defoliated extensively from the release point down the first part of the hill, and the observer is standing in the middle of the field where the beetles have not yet reached. And 2 years later, the beetles have completely defoliated. And this was taken in the 1950s and is used as, one of the most flagship indicators of the potential success of weed biocontrol programs. So sort of going back to Jen's presentation about how biological control works. So if we have an invasive plant, St. John's wort here, the populations in green may increase on the landscape and oscillate at levels that are often far above what we would consider acceptable ecological and economic damage thresholds. And the idea is once we bring in a biocontrol, a classic biocontrol agent, in this case, weed biocontrol agents, that reuniting them with the invasive plant will drive the populations of the invader down below damage thresholds over time. And this has been the case with St. John's wort, where the beetles and other agents were able to highly suppress St. John's wort on the landscape. However, we're getting to a stage now where we're 70, 80 years into the world's oldest biocontrol programs, and we have the opportunity now to ask, does biocontrol really stay effective in the long term? So here in British Columbia, as well as in neighboring regions of the US, in Eastern Washington, Montana, and Idaho, we've heard new reports of St. John's wort being out of control, often at high elevations with highly persistent populations of plants, rangeland being heavily impacted, and possible changes in conditions and efficacy of biocontrol. And so what is really driving these perceived increases? Are they true increases? Is biocontrol still working? So we've been beginning to get at those questions with our partners, the province of British Columbia. So if I bring you back, so this has been the state with biocontrol for St. John's wort, and it sort of matches our theoretical expectations, but it's not the only possible outcome for biocontrol. So if we're looking, say, here at two different sites where we might have biocontrol on the landscape, the red line being the point in time in which we're looking, we don't know, in the absence of long-term monitoring, we don't know where we are necessarily in the invasion curve. So at this first site, we might have populations of, say, St. John's wort that seem out of control, but maybe the insects aren't there yet. Or at another site where we have very low populations of St. John's wort, are the beetles effectively suppressing it? And so there's other possible options here. So if we look on the right, when we see a potential resurgence in a biocontrol program, so a peak of the invasive plant, the biocontrol agents are usually right behind, but we don't know if we're in a peak that's just part of a natural oscillation, or if we're in a peak that's sort of an anomaly, or what's not shown here, are we at the potential of having an invader that is no longer successfully controlled by biocontrol? And it really takes detailed monitoring at field sites to begin to understand what is happening when we see certain populations on the landscape that are at high density. So this is what Andreas has been working on, reevaluating the efficacy of St. John's wort in Canada. Here's a picture of Andreas diligently out in the field. And some of the questions that he's been asking that have been motivated by stakeholders here in British Columbia. Do St. John's wort populations vary in density, extent, and traits across geo-climatic gradients? Can variation in St. John's wort be attributed to variation in presence of biocontrol agents? And what influences the plant's plastic responses at different sites? Is it more related to phenology? Is it shown more in the coverage and abundance of the plants? What's driving these factors? And including looking at the phytochemistry of the plants, because as mentioned, they're highly toxic plants, not only to mammalian herbivores, but also to the biocontrol agents that have co-evolved with them and need to detoxify these plant metabolites. So our study area here is in Western North America in British Columbia, as I mentioned. This is an image from Invasives BC, our provincial database, which has immense records on the distributions of invasive plants in the province. And so there's many, many, many records of St. John's wort here. And this may overestimate the distribution because these are records. Many of these populations are under successful control, but some of them, as mentioned, are at a state where we're unsure where they are in the invasion curve and how effective biocontrol is. So Andreas is using 17 sites that are in interior British Columbia, and he spent a lot of time identifying sites that vary in the density and abundance of St. John's wort that have biocontrol agents. He's been investigating these geoclimatic factors, taking a number of measurements of plant traits, and looking at relationships with the biocontrol agents. So he conducted his study in 2024 from very early in the season before St. John's wort was leafing out all the way into winter. And we expected him to finish in October. However, some of the biocontrol agents are still very active late in the season, so his study was quite long. So he was looking at the phenology of the plants and other plant traits, geoclimatic characteristics of the sites, growing season length, and parameters related to the biocontrol agents. So I don't have a lot of time here to share results, and these are sort of hot off the press results that Andreas has just been pulling together for his thesis, which he's currently wrapping up the writing. And so looking at all of his analyses, he followed sort of similar steps using linear models, looking at how each response variable responds and correlates to the climatic, geographic, and biological factors. using growing degree days as the metric to see how the growing season length influences traits like plant phenology and presence and abundance of the insects, as well as looking at plant phytochemistry. I don't think that changed. I'm just going to go through this one to speed up. So for phenology, looking at the accumulation of growing degree days related to the phenological stage and responding to many predictors. So investigating phenology across these gradients. And so what Andreas found is that faster growth of St. John's wort plants occurred both with shorter and longer growing season lengths, whereas sort of at his mid-range of sites, plant growing season, is in the peak there. Sorry, I think I might just be getting oh, that's a question. So we'll keep that for later. He's looking at traits like time to flowering, shorter flowering time with longer growing season length, and faster timing to flowering when there are more insects present. So it's really interesting. It seems that when we have more biocontrol activity, this actually pushes St. John's wort to progress phenologically faster and flower and produce seed more rapidly. So how that has a long-term effect on populations is something we don't have the data for, but it's a really interesting finding. As well with fruiting, as I mentioned, slower fruiting time as growing season length increases, and the faster fruiting with more insect activity. So biocontrol agents definitely exerting differential impacts on plants when there's higher population densities of the biocontrol agents. The time for seeds to dehiss, so slower release time as growing season length increases. And time to biological death of the plants, so faster plant death with increasing insect damage. So this is one of the things we would expect or hope to see with strong biocontrol efficacy, is that as we have greater insect herbivory on the land, that it leads to faster death of the host plant of our invasive target. As well, though, longer plant survival with longer growing season length. So growing season length is related or associated with elevation. And so where we have longer growing seasons at lower elevation, plants live longer, and at higher elevations where there's less accumulation of growing degree days and temperature, the plants have a much shorter phenological window. And time to regrowth, longer time for plants to regrow as growing season length increases. So lots of parameters here on how the plants are responding to the environmental conditions, which is Andreas is continuing to work with these data to better tease apart how these parameters and biocontrol interact on the landscape. I just wanted to do a check for my time here.
You have a couple minutes still.
Okay. So Andreas as well has been looking at coverage and abundance of St. John's wort. Is plant coverage and abundance responding to these multiple predictor variables? And in the presence of biocontrol, and how biocontrol abundance varies with plant abundance. So Andreas has found that there's a higher absence level of plants with increased herbivory. So where we have higher abundance of the different biocontrol agents, we have fewer plants and lower plant density, indicating that biocontrol is correlated with lower plant density on the landscape. These insects do aggregate, they're not evenly spread. And so there can be really high aggregation of insects, leading to really low plant abundance in particular populations and within particular sites. Another chapter of Andreas' thesis is investigating how plant secondary metabolites associate with these different geoclimatic factors and with biocontrol and variation in the plant metabolomic profiles. So the prominent phytochemical is hypericin. It's very toxic to livestock and to the beetles, but the beetles can metabolize this compound, but it does have a life history cost for metabolism. So hypericin concentration, he found increases, and they increase non-linearly with eastness. So exposure to the sun, so aspect, has a role in hypericin and other plant metabolite performance. And so where plants are specifically on the landscape can influence the phytochemicals they produce and their interactions with biocontrol agents. There's other plant compounds like hyperforin that perform similar functions, and that beetles can metabolize in similar ways. But overall, looking at metabolic investment, Andreas's results indicate that plants are investing differentially between these different between these phytochemicals. However, most plants, regardless of their location, do show similar profiles overall, and some plants have increased amounts of hypericin. So working to continue to tease apart the role of these plant metabolites and how they interact with plant phenology, geoclimatic factors of the site, and biocontrol will actually be the subject of Andreas' PhD going forward, because we have a lot of sort of interesting leads here, but a lot more to tease apart. So just sort of in summary of the types of parameters that Andreas investigated, and in summary, from the work that he's done so far in his master's that will be continuing, shorter growing season length of St. John's wort correlates to faster plant phenology, that's an E, faster plant phenology, or shorter life history of the plant. Presence of biocontrol agents is associated with early plant reproductive investment. So plants are responding adaptively on the landscape to complete their life cycle much quicker in the presence of biocontrol. And insect presence also is associated with differential plant phytochemical concentrations. Plants with severe damage from biocontrol agents do die early, and that can lead to sort of more patchy spots in the landscape. But so far, Andreas hasn't been able to link that to population level impacts. More widespread insect damage correlates with fewer plants, and plants may change their strategies phytochemically in response to neighboring damage. We didn't show that data here today. Insects need to accommodate for faster plant phenological stages at sites that have shorter growing season lengths. So this is where we might start to yield some interesting results related to elevation, because when plants have to move faster through their life history at higher elevations because of shorter growing season length, the way that the insects are responding to that, they do need to adapt. So there's more work to be done there. Plants are able to shift strategies to defend against herbivory, but the insects are very good at compensating and building up high population levels. And variable plant toxicity, it is potentially a factor that's influencing variation, herbivory, and biocontrol effectiveness. So for questions that can go in the Q&A, Andreas is here, and he can put answers in a chat to me, and I can paste them back in when we get to that section. Thank you.
Thanks, Chandra. Let's shift over to Brittany Barker.
Well, thank you everybody so much for being here. So I'm going to be splitting this presentation with my colleague Fritzi Grevstad. We're both at Oregon State University. I'm at the Oregon Integrated Pest Management Center, Department of Horticulture, and Fritzi's with Botany and Plant Pathology. And we'll be focusing on phenological drivers and cues, a conceptual framework for thinking about how climate can interact with insect life cycles to affect voltinism and phenology. And to start out I started out with a definition of phenology. So we've heard in our previous two presentations the word phenology, so maybe everybody knows this already, but in case you don't, phenology describes the seasonal changes in the life cycle of plants and animals. So if we're thinking about a weed biocontrol system, the agent would probably be like an insect, as one example. So we have 4 different life stages. In this case, I'm showing a butterfly, but many biocontrol agents have these stages as well. So like a larva, pupa, an adult, an egg. And then the plant, which would be the weed, has its own phenological stages, including the seed, and then where there's vegetative growth and flowering, fruiting, and the fruit releasing seeds, and then finally senescence, wherein the plant dies. And so the voltinism of these organisms, that refers to the number of annual generations per year. So if an insect is univolting, that means it would have 1 generation per year. Temperature is a major driver of phenology as well as survival of insects and plants. And so I'm showing here this little silly cartoon. This is of a bee, just because that was the only little cartoons I could find. So I'm showing here a plot, right? So on the y-axis, we have developmental rate, 1 divided by the number of days. And on the x-axis, we have temperature in degrees Celsius. All right, so let's start at the bottom of this, or towards like the beginning of the x-axis. So below about 5 degrees C, this particular little cartoon bee experiences cold stress. So its development is halted at this point. Cold stress units are accumulating, and eventually the insect will die either from starvation or from the cold exposure itself. Once the temperatures rise above 5 degrees Celsius, the lower developmental threshold, developmental rates speed up. And so you see an almost like linear relationship between temperature and developmental rate. However, once we get up towards the upper developmental threshold at about 25 degrees C, that developmental rate plateaus, and then the insect begins to accumulate heat stress. All right. And that is a pretty steep drop off. Typically, like the cells will start falling apart. And that's why it happens pretty quickly. And then the insect will die. And I want to point out here that temperature is not the only driver of phenology and survival. So, for example, in the previous talk, we heard about how biotic interactions can also influence phenology, so the developmental rates. We also have photoperiod, which Fritzi will talk about in a few minutes here. So, decision support models can incorporate this type of information to predict phenology and survival. So, a phenology model predicts when to expect certain life stages of the organism that you're modeling. So, in a weed biocontrol system, you could have a phenology model for the weed. You could also have a phenology model for the insect. Here I'm showing a just kind of a basic insect life cycle. Again, we have a egg, larva, pupa, and an adult beetle. And then at the bottom part of the slide here, I'm showing you another graph. On the x-axis is dates, and on the y-axis is degree days. So degree days are a heat unit, and those accumulate over time when the temperatures are between the lower and upper threshold of the organism. In this case, this particular organism has a base temperature, has a lower developmental threshold of 50 degrees Fahrenheit. So we can predict when to expect certain phenological events in this organism with our model. So for example, here we have first egg hatch occurring on March 11th, and then further on we have like first instars and peak instars, and finally on July 2nd we have first adults. This type of information is important because we need to have releases of the biocontrol happening at the right time of year. We also might want to know when is the best time to go out and begin monitoring the insects or the weeds that we're interested in. We critically too, we can understand the synchrony of the insect and the weed host, right? So as mentioned by our previous speakers, that is really important, that phenological synchrony in the plant and the agent in order for the biocontrol system to be effective. Climate suitability models predict where establishment is more likely based on, well, there's multiple different types of climate suitability models, but one type, you can incorporate that information about cold and heat stress to be able to predict where survival may be limiting or not. And the reason that we care about climate suitability models is that we can make sure to release agents in areas that are more climatically suitable, where it's more likely to survive. Here I'm showing a rather basic climate suitability model. This is from a paper I published last year on Cerataphium bassicorn, which is a new biocontrol agent for invasive yellow star thistle. So areas where the CMI index is more red, that indicates those are areas that are more climatically suitable for this insect, which was derived from a single source location in Greece. And the bottom map shows areas where star thistle has been detected. The yellow areas are where star thistle is found. So we see there's quite a bit of overlap in some of these areas that are climatically suitable for the insect and where the weed also occurs. So we obviously want to target those areas for releases. Okay, so I really want to highlight, though, that we need dynamic modeling tools. So climate, as we all know, is changing very rapidly. We have rapidly increasing temperatures. The bottom maps are showing both spring warming and winter warming since 1970 in degrees Fahrenheit. So we see that the rate of warming varies across the contiguous United States. So for example, we see that there has been a particularly large increases in winter warming in the eastern half of the country. But overall, across all counties, it is both spring and winter temperatures are going up. The consequence is that we'll have shifts in phenology. We could have earlier like adult emergence, earlier egg laying, and also earlier emergence of the weed, and also shifting climate suitability. So areas that were previously too cold for the invasive weed, they're becoming warmer, so they could go into those new elevations. And it's the question is, will the biocontrol agent follow it? We also have changing plant-insect interactions, such as their phenological synchrony. All right, so this is an example of some of the phenological mapping that I'm doing, and I'm showing you an example for an invasive insect, not a biocontrol agent. I just want to point it out as an example of where I think we would be really great if we could start moving this direction, where we have these types of models available for more weed biocontrol agents in the Pacific Northwest and also just like the United States in general, more broadly. So I'm working on this modeling tool called DDRP. It integrates mapping of phenology and climate suitability. And I'm showing you maps for light-brown apple moth, which is an invasive pest that's present in California, and it's at risk to spreading to more areas. Areas that are dark gray are predicted to be too cold for establishment. The areas that have colors are where are the dates that egg laying is predicted to occur, the earliest date of egg laying. And I'm showing you maps for 2010, 2025, 2045, and 2070. And I just want to point out here that notice how we do have this decrease in cold stress. So we see increasing climate suitability across the Pacific Northwest. We also see phenology occurring earlier in the year. So compare 2010, it was May 5th. Fast forward to 2045 and 2070, it's April 7th. So almost a whole month earlier. And I also want to acknowledge my collaborator, Len Coop, in producing these maps and developing this model. Okay, so we do have a knowledge and implementation gap for biocontrol. So I'm suggesting that we need more of these like near real-time forecasts for weed biocontrol agents. One of the issues is we do have phenology models, but they're not operationalized. So we don't have them up on a website where someone can take weather data and like run the model for a location that they're interested in. Most climate suitability models for biocontrol agents use historical climate data, so like climate averages. But again, climate's changing rapidly, so we need to think about using more present day climate data sets. And probably one of the most important things is we lack data for model development. So we lack information on like developmental rates, such in like the thresholds, the heat stress parameters, cold stress parameters. But Fritzi, who I'm going to pass the talk off to now, she is going to talk about one of the models where we do have that information. We've developed these models for forecasting. So Fritzi, I'll go ahead and hand it over to you now.
I'm going to continue the topic of phenology models, but with the incorporation of insect photoperiodism. So let me first point out that pretty much all classical biocontrol agents undergo a significant change in climate when they're first introduced to a new region. So here's the knotweed psyllid as an example from a single source in Japan to a wide range of climates and latitudes in North America. And understanding a biocontrol agent's response to this type of climate change has been my research focus primarily, but you'll see that it can be relevant for other forms of climate change, such as long-term global warming and year-to-year variation. So one of the important ways that insects are adapted to their local climate is through the precise timing of their life cycle to coincide with favorable seasonal conditions. But rather than using the actual weather conditions as their timing cue, which can be highly variable day to day and year to year, a lot of insects use the more regular and gradual changes in photoperiod as their primary cue to align their life cycles. So this cue often takes the form of a switch between the reproductive and non-reproductive phases of the life cycle, when day lengths either lengthen beyond or shorten beyond a critical photoperiod. So this critical photoperiod is a trait of the organism, and it can vary among species, populations, and individuals. So in this short talk, I'll focus on a common type of insect life cycle that is multivoltine with a short day diapause response. A lot of biocontrol agents, including knotweed psyllid and the loosestrife leaf beetle, have this type of life cycle. These insects overwinter in the diapause in a state of diapause or dormancy. They emerge in response to warming temperatures and become reproductive. Then they go through one or more generations until a particular photoperiod sensitive stage senses that the day lengths have grown shorter than the critical photoperiod. And at that point, they enter diapause. So a key point that I want to illustrate today is that the temperature responses that Brittany described and the photoperiod responses can interact to cause interesting things to happen when there's a change in climate. So I'm going to give two examples. In the first case, suppose we have an insect that develops through the summer and reaches the photoperiod sensitive stage late enough in summer that day lengths are shorter than critical. And that gives them the cue to enter diapause. However, in a warmer climate, that insect would develop faster, reaching the photoperiod sensitive stage earlier when days are still long. And this insect would get the cue to remain reproductive and produce an additional generation. So this could be good for biocontrol because the second generation will provide added impact on the weed or the pest, but in some cases there might not be enough degree days left in the season for this generation to complete development. In this next example, suppose we have an insect that typically completes 3 generations before it enters diapause. In a warmer climate, this insect could develop fast enough that it gets the cue to enter diapause in the spring after completing just a single generation. So diapausing extra early like this means that it's going to miss out on those extra generations, and it's likely going to die because these diapausing insects are not able to survive the long duration into the following spring. And this is not just a theoretical result that happens in the model. It's been well documented in introduced populations of the tamarisk beetle by Dan Bean. So as you know, day lengths change with latitude as well as with time. And so the formula that we use to calculate day lengths in the model takes this into account. And I'll also point out that for any insect that we want to model, we need to quantify the photoperiod response. And this needs to be done experimentally by rearing at different photoperiods. And we've done this for 3 different biocontrol agent species. Okay, so what our model does is it applies the logic that I described earlier to every grid cell across the continent using available daily temperature data, and it tracks the phenology and the voltinism. So this is output for the knotweed psyllid. The map on the left shows the number of generations that newly introduced populations are predicted to attempt given this insect's photoperiod response. And this is whether or not there are sufficient degree days to complete those generations. The map on the right shows the degree of voltinism mismatch to the season duration. So the green areas are places where they would attempt up to 1 generation too many. That is, they diapause too late. The white areas are locations where they would diapause with good timing. And the brown areas are areas where they would diapause too early. And much of this brown area is actually where they would diapause after only just a single generation. We can also use the model to predict how established populations may be affected by future climates. The model here is parameterized for the loose-striped beetle, and specifically the population collected from the Yakima Training Center in Southeast Washington. For the current climate on the left, notice that there's a lot of white in this area, and that means that this population is fairly well adapted to the current conditions. But for the future climate on the right, you'll see that there's an expansion of the area that is colored light brown, where diapause might occur too early. So this could be detrimental to biocontrol because we know that in places where Galaricella diapauses early after 1 generation, purple loosestrife tends to rebound in the second half of the summer when there's no feeding pressure.
Just
Okay, now keep in mind with these predictions that
wrap up.
insects are likely to adapt their critical photoperiod, so we need to keep that in mind. Okay, so in closing, I'll add to Brittany's points about the value of phenology models. In particular, when we include photoperiodism, these models can provide improved prediction of voltinism and mismatch for newly introduced insects. They can also be useful for looking at effects from year to year, and can be used to explore possible effects from future climate change. So finally, and importantly, we can use them to understand evolutionary responses to climate change, including adaptation by way of the photoperiod response. Okay, I've left you with some references. We'd like to acknowledge Tyson Wetbrecht, Dan Bean, and Len Coop, and our funding sources. Thank you.
All right, so I'm thrilled to be here to share with you some research that I've been involved with recently looking at, surprise, surprise, impacts of climate change on biological control. So this is sort of taking sort of a global review approach rather than a targeted study. So before I kick this 10-minute talk off, I do want to highlight that this was a large collaborative team effort. So I want to acknowledge the 5 co-authors that I have on this project. And I've included the link to this publication at the bottom. If you can't access it, it is unfortunately behind a paywall. Just send me an email and I'm happy to send you a copy of this work after this presentation. So all of the other speakers in the session have done a really wonderful job at sort of setting up this background to biological control and climate change. And so I'm not going to really briefly talk about, right, for our particular purposes, definitions and things like that. So our definition that we use for biological control is that it's one organism, right, this biocontrol agent, that's being used to control another, and that other species we're going to call the target host. And as you heard right at the beginning of this webinar, right, these biocontrol agents go through this really strict selection process to make sure that they're going to be effective if and when they're released. And so part of that selection process wants to ensure that first that species is able to survive in the range that it's planned to be introduced into. If it's able to survive there, we want ideally these populations to become self-sustaining. So we want to make sure that these biological control agents can reproduce in that introduced range. And then obviously we want those agents to be effective at managing the particular target host for which they've been introduced to control. And so for our work, we were particularly interested in how climate change might impact these sort of different regions, or sorry, not different regions, different aspects of biological control agent selection and performance. And the other speakers in this webinar have done a really wonderful job at highlighting some of the many ways that we already have seen climate change impacting the survival, reproduction, and or performance of both biological control agents. But I also want to highlight that all of these impacts could also be occurring to that target host as well. Right, so with, for example, increasing temperatures over the wintertime, we can see increased overwintering survival either in our biocontrol agent or its host. We could see geographic range expansion again, either for that biocontrol agent or for that target host that it's attempting to manage. We could also see, as we just heard, right, shifts in the number of generations that we can fit into a particular year, and again, either for that biocontrol agent or host. And a lot of that is associated with shifts in developmental rates. And then lastly, we also see actual performance changes, some of those driven potentially by a loss of synchrony. Right? So maybe those biological control agents are no longer co-occurring on the landscape with the taxa that they were introduced to manage, or maybe there's something about their ecology that's changed. Maybe, you know, their feeding rate or parasitism rate is changing with climate change that might either increase or decrease their efficacy. And so for our team, we were really interested in looking at just the general state of the science. So what do we know, right? What systems do we have information about this relationship between climate change and biocontrol agents? And more importantly, where are the knowledge gaps? And so this is part of a much broader study that I did. And so there were 2 parts to this question. And just because I've only got 10 minutes, I'm going to really focus in on this empirical evidence. So what data do we have that's been published in peer-reviewed literature, specifically for some kind of climate impact on biological control agents? But if you're looking for some more specifics, go and have a look at that full publication. So to kind of get at the state of the science, what we did is looked for a variety of keywords, or we conducted a variety of keyword searches through the scientific database Web of Science. And so we did these keyword searches 3 times between June of 2022 and 23. And this resulted in just over 2,000 2,000 articles. And then we went through those 2,000 articles and removed any duplicates and removed any studies that actually, when you went and read the study, weren't really interested or didn't really examine environmental impacts on biological control agents at all. And so once we'd sort of done this data cleaning process, this left us with 194 unique studies. And these 194 studies, all of them had some kind of data that looked at an environmental variable and how it impacted a biological control agent. So I'm just going to launch right into the overarching results from this kind of overview. So one of the things that we were interested in looking at for these 198 studies is how were the data being collected in the first place? And so we grouped studies into 3 different categories. So they might have had a greenhouse or a lab component, a field component, and or a mesocosm. And most of the studies that we looked at are being conducted solely within some kind of lab or greenhouse setting. So most of the data that we have are coming from these very controlled environments, very few from field or mesocosm experiments, and just 8% of studies looked at multiple different types of settings, right? So maybe combining a lab study or a greenhouse study with a field experiment. So again, not super common to do more than one experimental approach. Next, we were interested in what kind of environmental variable these studies were looking at. So on the y-axis, I'm showing you all the different environmental variables that we were interested in, or that we found, I should say, across these 194 studies. And as you can see, 86% of those 194 studies examined temperature. So temperature is far and beyond the environmental variable that we have the most data for, followed by some kind of moisture variable and carbon dioxide. And nearly 3/4, or just over 3/4 of the studies that we looked at only looked at a single environmental variable. And so we know that environmental variables can interact with each other. Temperature and moisture are 2 of the most classic ones. And so at the moment, we don't have a great understanding about how these interactive effects of environmental variables might affect biological control agents. Next, we were interested in the types of biological control agents and the types of target hosts that these studies were looking at. And so if you look at the left-hand side, I'm showing you in blue the breakdown of biological control agents. And so we've classified them into 1 of 4 categories, and 85% of the biological control agents that we have data for are arthropods. And this is kind of unsurprising, as these are also the most commonly deployed biological control agents worldwide. If we look at what kind of species those arthropod biocontrol agents, or I should say those biocontrol agents in general, are being deployed to manage, we see that just over half of them are being deployed to manage another arthropod. So some kind of insect or mite, something like that. And then about 1/3 of the studies were coming from some kind of plant. So a plant being managed by a biocontrol agent. And then the rest were fungi, mollusks, nematodes, things like that. And then we extracted all of the information from these 194 studies, looking at the different responses in both the biological control agent, and then some studies also were examining climate effects in the target host as well. So we extracted that information too. And so on the y-axis for both of these graphs, I'm showing you the different kinds of responses that we were seeing data for. And so again, in blue, we've got the biocontrol agent responses. And so most studies are looking at how climate change is impacting the reproduction or development of biocontrol agents. Some measure of efficacy, and usually that's like a feeding rate or parasitism rate. And third, survival. And that kind of nicely mirrors those sort of 3 main categories that I talked about right at the beginning of my presentation. And if you look on the other side of the page, these orange bars, we've got the host responses. And we see some similarities, but a little bit different as well. So most of the data we see collected on host responses to climate change look at host survival, followed by reproduction and development. And then the third most common is looking at the actual abundance of that host. And then one of the other categories that we heard would be quite useful and interesting to kind of consider when looking at this sort of review of how climate change might impact biocontrol agents is considering what the application setting is for that biocontrol agent. So has that agent been released or is being considered to control? So what kind of environment is it going to be used in? And we found that 63% of those studies, the 194 studies, that biocontrol agent was being used to manage some kind of species that was based in an agricultural crop or farming kind of situation. So think, spider mites, aphids, or, tomato leaf miners. And that's maybe a little bit different than some of the examples that we've heard in the webinar today, which are maybe more of our second category, which is our environmental or biodiversity. So think, forest health or looking at maintaining or restoring natural ecosystems. And then our other two categories being human health, so thinking about, control of mosquitoes, for example. And then we did have a few that were completely enclosed in terms of a greenhouse agricultural environment. So obviously I have a lot more data that I could present, but I'm conscious of the time. And so I do want to just leave you with a couple of management and research implications that we think are important based on this work. So I've only presented to you the sort of biocontrol side of things, but we were also interested in how many of these studies look at both biological control agents and their hosts at the same time. And sneak peek, there's not a ton. So, prioritizing looking at both of these species because this is a relationship. And as I highlighted, right, a lot of these studies are based on just one environmental variable, primarily temperature. And so getting a few more studies looking at multiple environmental variables and trying to bring in more of these field experiments or gathering data in some kind of natural setting. So two sort of big takeaways from that is, obviously climate change is impacting some biological control agent performance, not all of them. But we could see that maybe this is something that we should be considering as we're selecting and going through this approval process for future biological control agents to ensure that perfect match continues into the future. And then thinking about our link here to RISCC, right, looking to build stronger researcher and practitioner collaboration. So if you're a researcher, right, looking to help gather this data, these baseline data on more species would be great. What species to work on. This is where those practitioners can come in, right? Tell us what are your high priority species that you're looking to study, whether that's a biocontrol agent or a host. Or maybe you have a field site and you could offer that field site or help us gather some baseline data so that we can start looking again in this natural field setting at how climate change over the next decade might be impacting either biocontrol agents at your field or the hosts. And so with that, I'm going to wrap up by acknowledging all of our funding sources. I am part of the Northeast RISCC, and so I've got their link here, and then I've also got a QR code to that publication in my email if you need help getting around that paywall. And with that, I'm going to turn it back over to Eva.
Thank you so much, Annette, and thank you to all of our speakers. Thank you as well to all of you who have stayed until this time. And with that, I'd like to move on to the fact that, as we've just seen, a lot of questions still remain around the impacts of climate change on biocontrol systems. And so with that, to better answer these questions, we're inviting researchers and practitioners who work in relevant fields, including biocontrol specialists, insect and or weed ecologists or physiologists, other researchers in that area, and climate change experts to join a working group. This working group will aim to identify and fill knowledge and capacity gaps around the impacts of climate change on biocontrol planning and efficacy, starting with weed biocontrol in the Northwest. And just For the bounds of what we're calling Northwest, we're using our Northwest RISCC definition, so that's Washington, Oregon, Idaho, and the surrounding states and provinces. Working group participation will entail attending 3 virtual meetings this spring, contributing to feedback on a working group report that will identify research priorities for Northwest weed biocontrol systems in a changing climate, and we'll have the potential to meet and form new collaborative research projects with folks who are in this group as well. So if this is something that interests you, we do have a sign-up form and interest form where you can let us know that you'd like to learn more. We'll start scheduling that first meeting. And so we have until next Friday to submit that interest form. I'll also have Darcy put that in the chat for you all. With that said, I want to thank everyone for being here. In addition to our speakers, we had other folks who were contributing to the planning of this, and Joey Milan's also been helping with Q&A. Please do add your questions into the Q&A box if you have any lingering questions about the content of any of our talks and or that potential working group. And in the remaining time, we'll answer those. Otherwise, thank you all for being here, and we look forward to sharing more with you as this moves forward.